26, he has mentioned the center of diversity. Center of diversity means where the maximum diversity do occur for a particular crop. That is the center of origin. As because in the origin of a particular crop in that particular place, the maximum number of diversity will found. Again, it is the secondary and primary center of origin. Vavilov originally mentioned the 8 primary center of origin, and second one is the secondary center of origin. What is secondary center of origin? Now it is that place where the variability is there, but not as much as of the primary center. Suppose the variation in the primary center of origin is 1000, for example, it's arbitrary value, and in the secondary center of origin it is 200 or 300 or 400, like this. So primary center of origin and secondary center of origin. Another terminology we generally use, that is the micro center. Now within a large center of the diversity, small area may exhibit a much greater diversity than the center as a whole. Suppose the center of origin is known the West Bengal, but more specifically in the Malda, Murshidabad, Nadia district are maximum variability you find. So the micro center of that is the Malda, Murshidabad and Nadia, but Malda, Murshidabad and Nadia, but the center of origin will be the West Bengal, like this. Now the quantitative and qualitative character, I think everybody we know about this terminology. Quantitative means the polygenic inheritance. So polygene or quantitative gene or minor genes means many gene with small effect, and the character is controlled by the quantitative gene is known as the quantitative character. Similarly, the qualitative character is controlled by the major gene or oligogene. So this character is known as the oligogenic character or major gene character. And the inheritance of the qualitative genes or qualitative character is known as the qualitative inheritance, and simultaneously the inheritance of the quantitative genes is known as the quantitative inheritance. So quantitative gene, quantitative inheritance, quantitative character. In contrast, qualitative gene, qualitative character, qualitative inheritance. So polygenic trait and oligogenic traits. Now polygenic governed by the several genes, here only few genes, 1 or 2. They each of the effect of the each gene is not detectable, with minor effect, small effect. And in case of oligogenic, the each gene is detectable, usually governed by the additive genes. Additive genes means I have already discussed. I'll coming in the next slides. What is additive genes and what is dominant genes and what is the epistatic genes? And it is governed by the non-additive genes. That means dominance and epistatic genes action are there in case of oligogenic. Variation is continuous, and here the variation is discontinuous. Then separation into the classes is not possible. It will be we will get the normal variation. You will the normal curve in case of polygenic trait. The phenotypic variation will be in normal mode. If we put the typical character into a graph, then we will find a normal graph. But here the variation will be discontinuous. That is either dwarf or tall, or long or short, like this. Separation into distinct classes is not possible. Here the separation into distinct classes are possible. Highly influenced by environmental factors, little influenced by the environmental factors. Polygenics are mostly influenced by the environmental factors. And statistical analysis is done by the mean variance and covariances. And here in case of polygenic trait, statistical analysis is based on frequencies and ratios. One example, that is the kernel color in wheat, Nelson L. in the year 1908. Nelson L. in the year 1980, he did this experiment from the cross between dark red and white seed parents. White seed parent, Nielsen observed the following results in case of wheat. So genotypes, parents, dark red or white, genotypes, that is capital R1, capital R1, capital R2, capital R2. Here all are in a recessive form. It is the medium red, when the 2 genes are dominant in each locus and 2 genes similarly, like this, the different types of in the F2 the segregation, after segregation, complete segregation, he is find the dark red, medium dark red, where the 4 alleles are dominant, the medium dark red, the 3 alleles are dominant, the medium red, the 2 alleles are dominant out of 4, and light red, the 1 allele are dominant, and the white all are recessive in condition. So this is the inheritance of kernel color in wheat. This experiment was did by the Nelson L in the year 1908 in wheat. QTL, another terminology you will find, that is the quantitative trait loci. Now, loci controlling the quantitative trait is known as the QTLs. You will find in several literature nowadays the QTLs. So that's why I am discussing with this terminology. The QTL means the quantitative trait loci. The term QTL was introduced by the Gelderman in 1975. Conceptually, a QTL can be a single gene or it may be a cluster of linked genes that affect the trait. And there are for example, I am giving you one separate example. Suppose for a single character, for panicle length, there may be more than one QTL in that genome, total genome. Suppose in chromosome number 1 there is one QTL which is responsible for panicle elongation. Again, in the chromosome number 7 there may be another QTL which is also responsible for the panicle elongation, and like this. So the QTLs for a single character may be much more, and that is distributed throughout the genome. And now the quantitative genetics. In the quantitative genetics, the component of variation we should know. The quantitative variation, there is the phenotypic variation, genotypic variation, and environmental variations. Variation I have already discussed, that differences among the individuals within the same species. So phenotypic variation, genotypic variation, and environmental variation. Fisher in 1918 divided the genetic variation into again the three categories. One is the additive variance, second one is the dominant variance and dominance variance, and third one is the epistatic variance. So I am not going in details with this part, but you should know the additive means just simple addition. There are 1, 2, 3, 4 genes. They are additive in nature. They contribute. When they come together, they will give you the better result. Dominance means is the recessive and dominance relationship will be there. And epistasis, it's the interaction between the genes. So these are the three types of genetic variation which has been classified by Fisher in the year 1918. Now heritability, another important, because I have already said that everything is based on selection, and selection will be effective if the heritability is high. Heritability, again, what is the heritability? The proportion of the phenotypic variance to the total to the genotypic variance. So genotypic variance divided by phenotypic variance into 100, that is the heritability, and it is known as the broad sense heritability, because heritability, again, it is of two types. One is the broad sense heritability, second one is the narrow sense heritability. If the heritability is low for a particular character, then the selection will not be effective. So you have to go in some other methods. We have to follow some other methods. So broad sense heritability and narrow sense heritability are two types of heritability found in plant breeding. What is broad sense heritability? Now it is the VG by VP. VG means genotypic variance to the total variance. Phenotypic variance means total variance into 100. Always heritability is represented by the percentage. So that's why it is multiplied by the 100. Heritability may be 60%, 80%, 4%, 5%, or 100%, like this. Of course, 100% heritability you never find, because to some extent some role of the environment must be there. So it is the broad sense heritability. And second one is the narrow sense heritability. As we have already discussed, the genetic variability, that is the VG, again can be classified in three categories. What are those? Now the additive, that is the VA, that is the variance due to additive genes. Then VD, variance due to dominance, and VI, that is variance due to epistasis. So again, this genotypic variance can be classified in three categories, that is VA, VD, and VI. So VA, VD, and VI. And narrow sense heritability is nothing but it is the proportion of the additive genetic variance, only the additive part, to the total part into 100. So always we will find that heritability calculated by the narrow sense is lower value than the heritability calculated by the broad sense heritability. So narrow sense heritability is estimated from the additive genetic variance. Already I have discussed this thing. Now the environment, another factor which affects the plant breeding. Environment, again, the two types. One is the micro environment, second one is the macro environment. Micro environment means the environment of a single organism, that's as opposed to the another growing in the same time and place. That's the physical attribute of the soil, temperature, humidity, insect pest, disease in your laboratory or in the field. So that is the micro. And macro means associated with the general location or the period of time, a collection of the micronutrients. So macro environment of the total Krishnagar and micro environment of your college. So this is the macro and micro environment. Then again, Allard and Bradshaw in 1964 classified environment into two groups. One is the predictable or controlled environment, and second was the unpredictable or uncontrolled environment. Control, what is the controlled environment in plant breeding? The fertilizer level, showing date and density, method of harvesting, high level of interaction is desirable. So these are the which can be controlled. The control is in your hand. But what is the uncontrollable environment? Now, season, rainfall, temperature, all these things you don't have any control. So these are the uncontrolled environment. So this environment has a greater role. This environment has a greater role in plant breeding. So this is all about the quantitative genetics and all these things. So quantitative genetics and quantitative and qualitative character. So we have to remember these things. So up to this, the mode of reproduction we have discussed, the quantitative terminology, and the qualitative and quantitative characters, the inheritance pattern, and all these things we have discussed. Now, directly I am entering the conventional breeding methodology, which are applicable in case of self-pollinated crop. So, choice of breeding method mainly depends upon four things. First one is your breeding objective. What is your objective? So, before going to start up any breeding methodology, you have to have a clear-cut objective. That is the most important. Without your objective, without your determination, you can't go for any breeding methodology. You can't go for plant breeding. So first one is the breeding objective of the crop. Second was you have to determine what type of pollination of that particular plant, whether it is a self-pollinated crop, or it is a cross-pollinated crop, or it is often cross-pollinated crop, or it is a vegetatively propagated crop. So based on the mode of pollination, you have to decide your breeding methodology, what you choose. Third was the mode of reproduction and the gene action. What type of gene action is there? Whether it is a quantitative in nature or it is a qualitative in nature, or whether it is a recessive gene controlled character or it is a dominant gene controlled character. So depending upon your gene action, mode of pollination, mode of reproduction, and breeding objective of your crop, the choice of breeding method have to be decided. Now, coming in the breeding methodology in self-pollinated crop, first one is the acclimatization and introduction. Already we have discussed. It is applicable in all types of crops. It is same as in case of cross-pollinated crop, or it is in case of vegetative propagated crop, or it is in case of self-pollinated crop. Second one is the selection. Two types of selection we do generally follow in case of self-pollinated crop. First one is the pure line selection. Second one is the mass selection. What is pure line selection? Pure line, what is pure line? Pure lines means is a population, is a offspring which are derived from a single homozygous plant. Remember it. The pure line selection, the pure line theory was given by the Johansson in the year 1903. So pure line, what is pure line? Now pure lines are the progeny of a single homozygous plant. Single homozygous plant. Progeny of a single homozygous plant is known as the pure line. So pure line selection, it is restricted to the self-pollinated crop only. So selection of the pure line, we have to isolate the whole material into the pure lines. So what Johansson in the year 1903 published his work, that is ultimately he have collected some beans from the market, and ultimately he got, after 7-8 years of selection, pure line selection, he got the several distinct classified category of the pure lines. And second one is the mass selection. Mass selection is the most primitive method. It is mainly used for purification of the variety. Suppose you have the variety, you have grown in the field, you have to rogue the undesirable plant or the plant which is not similar with the one, and the mass selection to occur. So these two types of selection is applicable in case of self-pollinated crop. One is the pure line selection, and second was the mass selection. Then third category is the hybridization. Pedigree method, bulk population method, backcross method, and multiple crossing. So pedigree method is the most common method and maximum used method in case of self-pollinated crop, and most of the 50% variety has been developed through pedigree method in case of self-pollinated crop till date. Pedigree means, it is the hierarchy, maintain the hierarchy. From the F2 onwards, you have to maintain, you have to keep record in your record book. So pedigree method, after 9 generation, you can easily detect from where this material has came. So this is the pedigree, maintenance of the pedigree. Bulk population method, again the bulk method is one of the method. It is similar to the mass selection, but only crossing is done in the preliminary stage. Backcross method, you know very well. The backcross method is for suppose it is used only to rectification of a specific character. Suppose in case of disease susceptibility, suppose one variety, for example, I am telling you, in one paddy variety, MTU 7029, the most popular variety, the mega variety of West Bengal, that is Lal Sarno, jetake boliye. What is told as Lal Sarno, MTU 7029, is susceptible to the sheath blight. So it is susceptible to the sheath blight. So in this case, we go for the backcrossing. If we get one source which is resistant to the sheath blight, which is resistant against the sheath blight, so that variety is used. That sheath blight resistant gene can be used as a donor to the susceptible one. That is MTU 7029. So backcross method, again, that character, that specific character which I would like to transfer, whether it is a recessive, controlled by the recessive gene, or it is controlled by the dominant genes, the methodology will be something different. And multiple crossing, that is by putting some more genotypes, we can bulk there, and the multiple crossing is another method of hybridization. Other methods, the multi-line variety are nothing but the isogenic line. Another terminology. What is isogenic line? The lines are genetically similar. 99.99% genotypes are similar in those lines. Suppose one multi-line variety having the 6 lines. So all the 6 lines are the 99% genotype, 99.9% genes are similar except one gene. It is mainly used for the disease resistance character. For example, in case of wheat, the multi-line variety has been developed. For rust disease, there are so many strains are there. So strain A resistant to a particular pathotype. Strain B is resistant to the particular pathotype of rust. Strain C like this. So every resistant genes is put in one line. So except that resistant gene, all genotypes, all the genetic architecture are similar. That is the characteristics of the multi-line variety. Single seed descent method, another method which is commonly used. Then hybrids and the population approach. And the population approach method. Single seed descent method means what we do in case of pedigree or in other method, bulk method, we harvested the whole plant. But in case of single seed descent method, single seed from single plant. Suppose in F2, there are 1,000 number of plants are there in the field. The say 1,000. So we will collect the 1,000 number of seed from one seed from single plant, and that will be bulked. And again in F3, that will be grown. In F3, again, single seed from single plant will be harvested and grown. And up to F5, it will continue. And after that, selection will be done. And the next, the methods, that is the preliminary yield trial or multi-location yield trial, will be conducted to release as a variety. So single seed descent method is one of the effective method. It's the modification of the pedigree method. And then the hybrids. Hybrid, you know, that is the F1. I will come later on, the inbreeding and hybrid in details, and population approach. Then these three methodology, mutation breeding, polyploid breeding, and transgenic breeding, these are the breeding methodology which are common in case of self-pollinated crop or in case of cross-pollinated crop simultaneously. So these are the breeding methodology. Mutation breeding, you know, the mutagens are used to treat the genetic material, the plant breeding material. It may be seed, it may be clone, it may be anything. So mutation, that is the different types of mutagens are there, you know. It may be ionizing radiation, it may be chemical. So anything, mostly in case of crop, generally the gamma radiation is mostly used, gamma radiation. So gamma radiation in BCKB itself, one gamma chamber is there. We have one project. We are working with the Bhabha Atomic Research Center nowadays, and one regional RNARC, one center has been established at Bidhan Chandra Krishi Vishwavidyalaya campus at Mohanpur, and we do regularly. The one gamma chamber is there, cobalt 90 gamma chamber is there, and we do regularly the treatment throughout the eastern zone from some materials generally coming from the Assam Agriculture University or Vishwabharati University or anything. So one gamma chamber is also with us. Then polyploidy breeding. Polyploidy, you know, poly means many, ploidy means level, many levels. So polyploidy breeding is another, and transgenic breeding I will come later on. So breeding methodology in case of cross-pollinated crop, introduction, acclimatization, these are the common. Then among the selection with progeny testing and without progeny testing. What is with progeny testing? Without progeny testing, what is what is progeny testing? The testing of the progeny ultimately give you the better result about the parent. So what type of parent you have selected, so that will reflect by your progeny testing. So testing of the progeny. So without progeny testing, the selection, the population improvement do occur, and with progeny testing, then plant to row or yak to row, anything, line breeding. These are the, then another method, recurrent selection. There are 4 types of recurrent selection. They are in case of generally followed in case of cross-pollinated crop. Simple recurrent selection with the GCA, recurrent selection with the SCA, and reciprocal recurrent selection. GCA, what is GCA? A general combining ability. What is SCA? That specific combining ability. So what is the difference between two? One very simple example I am giving you. Suppose you have developed 10 numbers of inbred. So you have 10 numbers of inbred that will be used for development of the hybrid. So with the 10 numbers of inbred, if we cross with a open pollinated OP variety, the parent is, that is the male is common. Male is common for those 10 inbreds. So in the left side, the female parents are there. Number is 10. The male parent is common. That is the open pollinated variety. It is known as the top crossing. So you will get the 10 numbers of F1s. And if we evaluate the performance of those 10 numbers of F1s, whatever the differences you will get, that is strictly due to the genotype of the female, that is the inbred. So by this way, we can identify which inbreds are good in genetic constituents. So suppose out of 10 numbers of inbreds, you find the 5 numbers of inbreds are there. Very good. So what happened? Now you can put those material, those 5 numbers of inbreds, and go for the seed production. And whatever the material you will find, that is known as the synthetic variety. And if we do not go for the GCA, without GCA, if we collect some inbred, good inbred based on phenotype, and put in the seed producing block, the resultant will be the composite variety. And what is SCA? The specific combining ability. Specific means 1, 2 out of 10 varieties, all permutation combination will go for crossing. 1 into 2, 1 into 3, 1 into 4, likewise 1 into 10. Similarly, 2 into 2, 2 into 3, 2 into 4, 2 into 5, like this. So by going each specific crossing, the specific combining ability is tested, that which combination is giving you best results. So out of 10, there will be how many numbers will be there? The factorial n by 2, that is without reciprocal, and factorial n is with reciprocals. Show specific combining ability for specific hybrid. Show heterosis breeding. In case of heterosis breeding, we generally consider the SCA, specific combining ability. Show suppose inbred number 1 is crossed, if it is crossed with the inbred number 9, that gives the maximum result. So this 1 and 9 combination will give you one hybrid. So this is the specific combining ability is used for the hybrid development. So general combining ability and specific combining ability, these are the very important part for the cross-pollinated crops. And the fourth one is the reciprocal recurrent selection. Backcross method, again common. Then I have already discussed heterosis, synthetic breeding, composite, distant hybridization. Distant hybridization means it may be interspecific hybridization or it may be intraspecific hybridization. Intraspecific hybridization, that is the hybridization among the varieties within the species like this, and intraspecific hybridization is known as the distant hybridization. Interspecific or intergeneric hybridization, that is known as the distant hybridization. Again, polyploidy, transgenic breeding, and mutation breeding. Transgenic breeding I will discuss later on. Then breeding method in vegetatively propagated crop. Introduction, acclimatization, it is common. Clonal selection, clone is the vegetatively propagule in case of vegetatively propagated crop. Then hybridization we can do if the sexual hybridization, sexual mode of reproduction is sexual. We do hybridize and then maintain the through clones. So hybridization, mutation breeding, heterosis breeding, and tissue culture, micropropagation. Micropropagation again can be followed in all the crops. In case of self-pollinated crop, the tissue culture can be followed. In case of cross-pollinated crop, the tissue culture can be followed. And in case of vegetative propagation, it is widely accepted. It is commercially exploited nowadays. Sure. What happened? Now, in case of if we produce the double haploid, if we produce suppose either pollen or egg cell, mainly from pollen, if we double the pollen, then 100% homozygosity you will get in a single generation. So 100% homozygous plant you may find through double haploid techniques. So tissue culture is another technique. It's the synopsis. What we do in case of micropropagation. Micropropagation is also known as the tissue culture. Many genetically identical plants are produced from one plant. It's a rapid, it's a asexual method of reproduction. Steps: first you have to select the explant. It may be apical bud or any other tissue. Depending upon the tissue, the name can be changed. Then establishment of the explant into culture medium. Then multiplication. The explant gives rise to a plant, a single node, produce a shoot within the 4 to 6 weeks. Then subculturing. Then root formation by maintaining the auxin-cytokinin ratio, the root formation, then shoot formation, and ultimate part is the hardening from the tissue culture lab to the field. So this is the in brief, the synopsis of tissue culture. Now, coming to the next heading, that is the inbreeding and heterosis. What is inbreeding? The most important part in plant breeding. The inbreeding means within breeding. Within of what? Within of relatives. Within of genetically relatives. If the breeding do occur, then inbreeding will come. So inbreeding means, and inbreeding depletion will come. So what is inbreeding? The mating among the individuals who are genetically related. Different forms of inbreeding are there. In case of plant, selfing is there. Selfing is the highest form of inbreeding. In case of animal or unisexual, the selfing cannot be possible. So where the selfing is possible, that is the highest form of inbreeding. That is both the male part and female part are similar. So that is the selfing is the highest form of inbreeding. Second one is the sib mating. Sib mating means brother-sister mating. Parents are common. Suppose we do cross A into B. The F1s are, we found some F1s. If the crossing is done between those F1s, it is known as the sib mating. That is the brother-sister mating. Brother-sister mating. Then half sib mating. Either sorry, half submitting. The one father or mother is common, either father or mother is common. That is the half. That is the step brother, step sister with the brother. That is half mating. And mating among other distantly related relatives is another form of inbreeding. That is the fourth form of inbreeding. So there are four types of inbreeding. First one is the selfing. Second one is the sieve mating. Third one is the half sieve mating. And fourth one is the mating among the other distantly related individuals. Inbreeding, what is the result of inbreeding? Result of inbreeding is the inbreeding depression. Inbreeding depression means loss of vigor and fertility in the next progeny. Why do follow? We are coming. So degree of inbreeding depression, high inbreeding depression, moderate inbreeding depression, and low inbreeding depression, and absence of inbreeding depression. In some plants, you will not find any type of inbreeding depression. Where? In case of self pollinated crops. So effects of inbreeding. Now why the inbreeding depression do occur? Now appearance of the lethal and sublethal alleles. Mostly lethal and sublethal alleles are lying within the genome as a recessive forms. Within the genome as a recessive form. So when the inbreeding do occur, then appearance of those recessive form occur as a homozygous condition. When the recessive alleles are present in the homozygous condition, it will be lethal or sublethal. The destructive effect, deleterious effect will be no. Second one is the reduction is vigor, reduction is reproductive ability, separation of the population into the distinct lines, then increase of in homozygosity, reduction in yield. So these are the effect of the inbreeding. Now inbreeding depression, reduction of the survival and fertility. It is known as the inbreeding depression, and it is measured through the inbreeding coefficient. What is the inbreeding coefficient? The formula is inbreeding coefficient that is represented by F, He minus H0 divided by He. He means expected heterozygosity, Ho means observed heterozygosity divided by expected heterozygosity. If the value of F is 0, so there is no inbreeding. That means expected heterozygosity is equivalent to the observed heterozygosity. If it is 1, then complete inbreeding is there. If it is in between, then degree of inbreeding vary. It is whether it is a moderate or it is a high or it is a low. So high F means significance of inbreeding. So inbreeding depression is measured by the inbreeding coefficient. So what is inbreeding? The mating among the individuals within the relatives. So first one, what is inbreeding depression? The reduction of the survival and fertility is known as the inbreeding depression. Is the result of the inbreeding. What is inbreeding coefficient? Now it is the coefficient by which we can measure the inbreeding depression. So inbreeding coefficient, it is known as the. Now the heterosis. Now I am giving you one example. If we understand this example, then so many things will be clear. Suppose there are two parents. Listen carefully. There are two parents. One parent is A, second parent is B. The value of A is 10 and the value of B is 6. It's an arbitrary value. Just assumption. So one parent A, the value is 10. Second parent B, the value is B. We do cross between A and B, and you will get the F1. Now suppose if we get the value of F1 is exactly 10, what you can say? It is a dominance. Dominance of what? Dominance towards parent A. If we found the F1 value is 6, what is that? Again complete dominance. What type of complete dominance? Complete dominance towards parent B. If we consider the value of F1, if we get the value of F1 is 8, that is in between two parents, 10 and 6, 8, exactly 8. It is the case of codominance. Codominance means both the genes are contributing and ultimately you will get the mid-parental value. So it's a case of codominance. If the value is in between 8 and 10, just imagine If the value of F1 is in between 8 and 10, it's an incomplete dominance towards parent A. If the values of F1 lies between 6 to 8, then again in case it is an incomplete dominance towards parent B. And if the value is greater than 10, it is heterosis. And more specifically, you can say it is heterobeltiosis. Heterobeltiosis means when the value of the F1 is exceeds the beta parental value, it is known as the heterobeltiosis. And if the value of F1 is less than 6, again it is a heterosis. Again it is a heterosis, and it is a negative heterosis. And sometimes the negative heterosis is very useful, very much useful in plant breeding. What are those? For example, in labs, you know, there is a toxic material of BOA. BOA. Previously, it was known as the ODAP, ODAP. It is a neurotoxin. If the in a 1F1, the BOA content is less than value, so it is desirable. So negative heterosis is also desirable. In case of cereal, the height, cereal, not in sugarcane. In sugarcane, if the height is negative, it is undesirable. But in case of cereal, if it is dwarf in nature, it is desirable. So there are some specific characters where negative heterosis is useful. And otherwise, in most of the cases, the superiority of the hybrid is expressed in case of biomass, seed, yield, speed of development, and like this. So heterosis can be calculated in several ways. Sometimes heterosis is calculated based on the mid-parental value. With that specific example, the value of A is 10 and value of B is 6. The mid-parental value is 8. If the F1 is calculated from the baseline of 8, it is the mid-parental heterosis. If the heterosis is calculated from the baseline of 10, it is a beta parental heterosis. If it is if the heterosis is calculated in a commercial variety, suppose one variety is very much popular in a particular area. I would like to introduce one hybrid in that area. So I have to compare the heterosis with that commercial variety. So that is known as the standard variety. So that is known as the standard heterosis or economic heterosis. So three types of heterosis are there. One is the mid-parental heterosis, second one is the beta parent heterosis, and third one is the standard heterosis or economic heterosis. Economic heterosis. Genetic models of heterosis. The dominant hypothesis is there, over-dominant hypothesis is there, and pseudo-dominant hypothesis is there. So three type of pseudo-dominant hypothesis is there. That is the epistasis model, and dominant hypothesis, that is each and every locus is dominant. So suppose in this case, the parent one, the A locus is recessive in nature, B locus is dominant in both the cases. P, these are the inbreds. P2, the A locus is dominant in both the cases. It is dominant allele. Here it is. If we do cross between these two, it will be the heterozygous condition. In both the locus, one dominant and one recessive. That means the result will be just double of this P1 and P2. So this is the dominant hypothesis. In over dominant hypothesis, suppose for example there are 5 alleles of gene A1, A2, A3, A4, A5. As per over dominant hypothesis, the more distant alleles, that is A1, A5, will be more heterotic than A1, A2. So this is over dominant hypothesis, and pseudo dominant hypothesis, that is the epistasis heterosis. Now, in brief, I will discuss some burning issue. Now, genetically modified organism, GM crop, because though it is not directly related to your syllabus, but I think some idea you should keep in mind, as because these are the burning issue nowadays. What is GM, genetically modified organism? Should I discuss? Hello, Pinto sir? Okay, what is GM? Hello. Hello.
Yes, sir, you can discuss about GM crops.
Hello. I am audible now.
Yeah, you're
Am I audible?
audible. Yeah. Yes, sir.
Okay. 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 What is GM? Genetically modified organism. Developing a crop of conferring or deleting a specific target traits to the host crop by insertion of a definite DNA sequences from any source. I have already told, I have already discussed that source must be genetically unrelated. So that is the GM. So this is the history of GM. First transgenic plant produced in 1985 in case of tobacco. Then particle bombardment method in 1988. GM crop considered substance equivalent to the hybrid varieties 1992. 1994, flap sap of tomato is released. Then herbicide and insect resistance crop have approved by the cultivation in 1996, of course in USA. 1998, mostly GM crops started by the multinational company Monsanto. Then GM food is dangerous, is declared by the UK. 1919, GM corn excluded from the baby food, and Greenpeace started the anti-GM campaign. And 2000, the golden rice developed, and later on, you know, the GM rice is ready. GM BT brinjal, a lot of controversy has been developed on BT brinjal. Then the BT cotton is a success story is there. BT cotton, and nowadays the most of the cotton crop are the BT cotton. Most of the 80% crop is controlled by the BT cotton itself. Genetically modified of the transgenic. What are the process? Now, incorporation of the gene of interest. What type of gene you would like to? For example, in case of BT cotton, what happened? BT cotton, the major problem is the cotton bollworm. The cotton bollworm is a lepidopteran pest, which can destroy, which can get entered into the boll. There is the fruit of the cotton, and ultimately the quality of the cotton is deteriorated. So cotton bollworm is the major pest of cotton. So BT, Bacillus thuringiensis, that is available. It is one bacteria. It is the gram-negative bacteria found in soil. So scientists are isolated the BT gene from this bacteria, and it has been transferred successfully to the BT cotton. So what happened? Now, when this BT gene from the Bacillus thuringiensis bacteria is transferred, there is a gene of interest, is the BT gene, has been transferred to the BT cotton. The whole cotton plant, the BT gene, after getting insertion, they are active in nature. If the lepidopteran pest feeds, then inside the guts of that insect, cry1 protein has been developed, and ultimately the insect dies. So this is the mechanism of BT cotton, and it is now a successful success story. BT cotton has been released by the government of India. Except BT cotton, no has been released by the GEAC, Genetic Engineering Approval Committee. BT cotton, BT brinjal, you know, lot of controversy was there few years ago. Jairam Ramesh, the then minister in charge, he was at Kolkata Bose Institute. I was there also for taking the public opinion, and ultimately the BT brinjal is not developed, not released for cultivation by the farmers. So this is the process. This is the end process. Co-cultivation of the agro. Achha, just 1 minute. Gene of interest. What is the how transgenic plants has been developed? Now, gene of interest, that is the identification, cloning, and sequencing. This is the first step. Then standardization of the regeneration media and hormone for the desired host plant. Third one, the interest of the gene of interest into the tDNA, transfer DNA. Border of the agrobacterium. In the agrobacterium, a soil-borne bacteria is a gram-negative bacteria. Then agrobacterium tumefaciens. So inside the cell of the agrobacterium tumefaciens, that is the tDNA. In the tDNA, the gene of interest, that is the BT gene, has been incorporated. Then co-cultivation of the agrobacterium culture and the plant part explant. then regeneration of the plant on the pre-standardized regeneration media, then selection of the plant with gene of interest, whether that BT genes is present or there, we have to justify, then field testing for checking the desirable performance. This is the, in brief, the points by which this is the plant transformation procedure in nature. Agrobacterial tumefaciens is there. TI plasmid, the bacterial genome is there. tDNA. So the transfer DNA has been incorporated TI plasmid. Then plant co-cultivation. Then the crown gall. It is the symptom. That means this tDNA has been transferred to the plant and ultimately co-integrate TI plasmid, TI cell, and it has been screened through the antibiotic, kanamycin resistant gene, and ultimately the cell of the transgenic plant is developed. I am not going in details. Some issues frequently raised against GM crops: the potential for gene transfer from genetically modified plant to gut into the flora and mammalian cells, the safety of the antibiotic resistant gene as a marker, potential allergenicity of the GM food, and non-target damage of the unintended damage. For example, in case of BT cotton, it is active against the lepidopteran pest. There are so many lepidopteran pests which are beneficial in nature. So if they visit in the BT cotton field, they will also destroy. So it can damage the non-target individuals also. BT cotton, I have already discussed in detail. Another technology of terminator technology. Just for your information, it's a production of the suicidal seed. That means the seed you will produce, that will be sterile. Again, the multinational company, they have developed this initially by USDA and multinational company, but throughout the world, nowhere it has been introduced. Why? Because the seed, the suicidal seed, some genes, sterile genes for sterility has been introduced in those seeds. In that generation, it will germinate, you will get the yield. And after harvesting, if you would like to sow again, that's it will be sterile. So it's a terminator technology. It's a suicidal seed production technology. So nowhere it has been accepted. And India and Brazil have already passed the national law to prohibit the technology. Golden rice, another golden rim project. It has been developed by the German scientist Dr. Bayer and Swiss scientist Dr. Ingo Portraistas by transferring one gene from Arwenia bacteria and two genes from daffodils into a japonica variety Tepai 309. Golden rice project was sponsored by the multinational Syngenta. Syngenta, American-based company. And golden rice is the beta-carotene, precursor of vitamin A, enriched rice. It contains beta-carotene, 160 microgram per 100 gram of rice. Though radish leaf contains 4,500 microgram per 100. So, but it's a research activity. It's a unique one. The beta-carotene, geranyl diphosphate, then phytogen phytone synthesis enzyme activity. This has been produced the phytones and the phytoene disasterase, G carotene, Z carotene, then Z carotene disasterase, the lycopene, and ultimately alpha carotene and beta carotene. So this is the mechanism of the production of the golden rice. Facts is that GM crop prohibitively expensive to develop. Traditional plant breeding is still the best option of genetic improvement in crop plants, and GM option chosen only when the gene of interest is not present in the same species and when the genetic improvement cannot be done without traditional plant breeding method. GM crop are released after food and environment safety assessment. No other food or crop improvement research is totally infected. Now, last part, 2-3 slides I will show the recent activity in the plant breeding, the molecular breeding and transgenesis, emerging the two potential tools for genetic enhancement of our crop. Both are driven by the genomic information. Already I have discussed the marker assisted breeding and the GM, that genetically modified crop. I have discussed about the GM. Now the marker assisted breedings, that means molecular marker are used. There are so many markers. Markers can be classified as a morphological marker. Suppose one character is associated with a morphology, for example, pigmentation, specific pigmentation. So that is the morphological character by marker. Then molecular marker, physiological marker. Like this, there are so many types of marker. The most effectively used marker and most precise used markers are the molecular markers. So by using these molecular markers, marker assisted breeding development is going on. And nowadays, for precision breeding, marker assisted breeding is the most important tool for us, for a plant breeder. Omics, just know the terminology. Omics technology are primarily aimed at the universal detection of genes. When it is omics technology is for genes, it is known as the genomics. When it is restricted to the mRNA, it is known as the transcriptomics. Because you know, I don't know whether the students who are participating is read the protein synthesis. I think you have read. So protein synthesis, you know, DNA to mRNA, mRNA to peptide or protein. So DNA to mRNA, that is known as the transcription, and mRNA to protein, that is known as the translation. So depending upon the steps, this omics technology is utilized. When it is restricted to the whole genes, it is known as the genomics. When it's restricted to the only mRNA from DNA, the mRNA produced, mRNA within a cell, produced through DNA. So it is known as the transcriptomics. When it is restricted to the translational product, that is mRNA to protein, it is known as the proteomics. So when it is the metabolites, different types of metabolites, it is metabolomics. So in a specific biological sample. The main differences between genomics and proteomics is the genomics is the study of the entire set of genes in the genome. Genome means the whole set of genes of a cell, whereas proteomics is the study of the entire set of proteins produced by the cell. Similarly, it is transcriptomics is the total mRNA inside the cell, and metabolomics means the entire metabolites inside the cell. So these are the new some terminology. So genomics might provide new avenue for discovery of new genes, QTLs, quantitative trait loci, and alleles. So possibility of genetic environments of our crops. We have started from Arabidopsis, only 120 MB, 25,490 genes are there, and then Drosophila, then elegans, then rice. Then nowadays, more than 400 genomic crops have been sequenced. Total sequenced has been there. So genomics, that is again classified structural genomics, functional genomics, comparative genomics, transcriptomics I have already discussed, proteomics and metabolomics like this. Sure. Thank you.
Thank you, sir. Thank you, sir, for this nice presentation of yours. You have enlightened us on the various topics of plant breeding. You talked about the types of pollination, the different modes of reproduction, about the transgene, the cisgene, about male sterility, apomixis, apogamy, etc. The basic principles of breeding are selection, and you talked about the variations which must be can be heritable or it can be non-heritable. They can be controlled by genes or they can be controlled by the environment. You said that early generation selection is a must for plant breeding, and the selection can be based on phenotypes, it can be based on genotypes, and the problem lies here when you select a phenotype, because it is controlled by the genotype, the genes, as well as it is controlled by the environment as well as their interaction. You also discussed about the different modes of plant selection, of plant breeding in self-pollinated plants. You talked about the mutation breeding, the you talked about hybridization. You talked about the heritable variations. You also talked about acclimatization, the micro centers, and finally you talked about polyploidy, transgenic breedings as well. You talked about the breeding methods in vegetative propagated crops, in cross-pollinated crops, as well as you discussed about the GM crops, that is the genetically modified crops, their gene interactions which are going on over there. And lastly, you talked about the BT cotton, the BT brinjal, the ethics behind them, and also you talked about the mechanism of BT cotton formation. And finally, you talked about the termination technology, the golden rice which has been introduced by Syngenta, as well as you also said that the traditional breeding is preferred than the non-conventional and modern breeding techniques. You also, lastly, you talked about the marker-assisted breeding. Thank you, sir. Thank you for this nice presentation of yours.
Thank you very much.
Okay. Okay, sir. Okay, sir. Now, over to Dr. Rituparna Kundu Chaudhary for the questionnaire session. Students, you can have questions too. Dr. Rituparna Kundu Chaudhary? Dr. Rituparna Kundu Chaudhary? Can you hear me? I think the microphone is mutated. Dr. Rituparna?
The microphone is open. Microphone is not muted, unmuted.
Yeah,
It is showing
unmuted. I think the connection is not okay.
Maybe.
Students, you can carry on questions if you have questions. Right now, sir is present. Yeah, she is there. Dr. Rituparna Kundu Chaudhary.
Yes, yes.
Yes, thank you, Dr. Bhattacharya. Thank you, Dr. Bhattacharya, for your elaborative and lucid explanation. I think
Dr. Rituparna, please speak. Some speaker should be in front of your mouth. Very low sound is coming.
Yeah.
Is it okay now?
Yeah,
now,
it's okay
it's
now.
okay.
Okay.
Now it's
Thank you, Dr. Bhattacharya, for your elaborative and lucid explanation. Our students are highly benefited from this thorough discussion. And this, on behalf of the students and all of our teachers, once again, I extend my gratitude, sincere gratitude for giving us your valuable time and knowledge. And I can see in our chat box that there is no question. So
that means
Is it that they have understood?
they have understood thoroughly. They are very much pleased with your discourse.
Dr. Rituparna, if there is no question, so there will be two options. One, nobody understand anything, and second, they understand
No,
everything.
I don't think so. I don't think so, sir. Your explanation was so high in quality. So I think that it was a really authoritative discourse. So I would like to conclude this session and would like to advance to our next session. That is, I would like to call up Dr. Pintu Banerjee, our head of the department, to confer the vote of thanks. Dr. Banerjee, please. Dr. Banerjee?
Please unmute yourself.
Are you there, Dr. Banerjee? It's time to call for the vote of thanks.
Yeah.
Okay.
Good evening. I feel proud to express vote of thanks for this state level webinar entitled Plant Breeding and Top Plant Improvement Strategies and Overview. It's my great pleasure and opportunity to convey my sincere gratitude and heartfelt thanks to the speaker, Dr. Prabir Kumar Bhattacharya of BCKV, who is very busy with his research work and has very nicely discussed the topic plant breeding and crop plant improvement, understandable to the audience from any discipline. He has discussed this topic very elaborately and part by part, starting from the self-pollinated crop to the cross-pollinated crop, and by the hybridization, mutation breeding, and also the modern techniques like the gene transfer method and also omics and other ethics related to GM crops. So many thanks to Dr. Bhattacharya. Now
Thank you
I
very much.
convey my sincere gratitude and thanks to our Honorable OIC Sir, Dr. Shobhan Nyogi, who is also busy with his administrative work for the distribution of mark sheet of third year BABSc students. I extend my heartiest thanks to the IQAC coordinator, Dr. Amaresh Mondal, for his constant inspiration and support. I want to acknowledge our advisor, Dr. Kalidas Das, for his creative and constructive suggestion. I want to convey my thanks to the coordinators, Dr. Sutapa Sannal and Dr. Kaushik Sarkar, who very nicely coordinated the program and communicated with all sectors. I want to convey my thanks to the convener and joint convener, Dr. Shormista Maiti and Dr. Rituparna Kundu Chaudhary, respectively, who are the pillars of our Department of Botany. They always help me to research the departmental activities in every way as and when required. I want to thank our departmental colleague, Dr. Sumanjana Khatua, an organizing member of this webinar, for her active participation, support, and cooperation. Now I want to convey my heartfelt thanks to Dr. Krishnendu Sarkar from the Department of Physiology of our college. And as he has acted as the technical advisor of this webinar. So Dr. Krishnendu Sarkar and Dr. Kaushik Sarkar actually created the Google Meet link and all other technical things of this webinar. Without their support and effort, we could not be arranged this webinar. So many thanks to them, and we are really indebted to them for organizing these webinars. We have organized at least 7 to 8 webinars. All are with their help. So On behalf of our department, I personally indebted with them. Thanks are due to all my colleagues and supporting staffs who are present here. Last but not the least, I extend my sincere gratitude and thanks to all the participants and beloved students who are watching and listening this webinar since 6 p.m. to make this webinar a grand success. So, thank you once again. Thank you, sir. Thank you, Dr. Bhattacharya. With your busy
sir.
schedule, you have delivered this lecture, and our students have benefited a lot. Thank you. Thank you once again. Thanks. Now this seminar is over here.